Can you like switch it off and switch it back on?
Video settings. 1 minute.
Also, yes, one more thing I've been meaning to ask you. Have you been facing any difficulties regarding the assignments and the tests?
Yes, ma'am, it's not opening.
It's
I've
not
been
opening.
trying, yeah. I've been trying to open it, but there is nothing.
Okay, so you have the app for that?
I don't have the app, but it's on my Safari. It opens in the Safari.
Okay, so you open it on a website.
Yeah.
Okay, like I think there's an app available also. So you can
Okay.
download the app and maybe it
Okay.
would work like that. Okay.
Okay, ma'am.
Yeah. And also if you are facing any kind of difficulties of that sort, that you're not able to open the test or you're not getting notified about anything,
Yes.
then please let the team know about it.
Okay.
Okay.
Yes, ma'am.
Hmm.
1 second, ma'am. I'll join from another device.
Okay. Am I audible? Just 1 second. All right. So we were talking about cryopreservation. Now see, cryopreservation is the method by which we can store the pollen grains for future use. Like for example, if we do not have any use for the pollen grain as of now, or if let's just say that you want to develop a plant of your own. Okay, so what you will do with it, you'll try to save the pollen grain so that you can use it later on. Okay, so it is the method of storage of pollen grains. That is what we call as cryopreservation. Now, you know that for the storage of any living thing, we have to put certain chemicals so that it doesn't die, isn't it? So same way, these pollen grains, they are stored for years and years in liquid nitrogen under minus 196 degrees Celsius. That is the temperature. And what is used? Liquid nitrogen is used for the preservation or for the storage. Now, such stored pollen can be used as pollen banks, just like how, for example, you go to a florist and you ask them for a specific seed, they'll have it, isn't it? You ask no matter whichever seed you want, they'll have it. Why? Because they have a storage of that seed, isn't it? But same thing when we do it with the pollen grain, so that we can, you can say we can breed the new variety of plants. So in that we have the pollen bank, just like how seed banks are there. Okay, the only two things that are important is like the one line that is the most important is like what is it called? So it is called as cryopreservation. And how is it done? So it is done by storing the pollen grains in liquid nitrogen at minus 196 degrees Celsius. Got it? And it is used in the crop breeding programs. These are the programs in which humans like us, what we do is that we take two superior qualities of the plant and we bring them in one plant. For example, there is a plant A that has really tall height. It's really tall, but it is not able to make more fruits. Then there is plant B that is making a lot of fruits but is not tall. So we take one good quality from here, one good quality from here, and now the offspring plant will be tall as well as it will make many fruits. Okay, so that is a breeding program. So for that, obviously some plants are seasonal, isn't it? But if you want to breed the plant in the non-seasonal type of condition, so we'll do the pollen, like we'll use the pollens that have been stored already. Clear? So with this, we have actually completed the male part of the reproductive area. Now coming on to the female gametophyte, that is gynoecium. What is the one unit of gynoecium called as?
Pistil and carcel.
Carpel. Carpel. Pistil or carpel. So it is the female reproductive unit of the flower, and the one unit of the gynoecium is what we call as the pistil or carpel. Now, whenever a flower has just one pistil, we call it as monocarpellary. Mono means one. Carpellary refers to the condition wherein there will be only one pistil present. Okay. And whenever there will be more than one pistil present, so we'll call it as multicarpellary. Multi means many, multiple, and carpellary again refers to the pistil. So we can say that when, oh, I've written gynoecium may contain single pistil. So we'll call this condition as monocarpellary. And when there are more than one pistil, we call it as multicarpellary. Clear? Also, dear, one more thing. If you have any doubts anywhere, do ask me, okay? Don't hesitate. Anything comes to your mind, even if it is tiny, I'm telling you from day one, even if it is very tiny, then also you ask, okay? Now, let us see. You know that the cells in any organism's body, whether it is a eukaryote or it is a prokaryote. So what happens? The cells, there are two types of cells in the body. The first one is the somatic cells, which is the body cells, meaning the cells that are not taking part in reproduction. Okay. For example, like our skin. Our skin doesn't take part in reproduction, right? It doesn't make gametes. So it is a vegetative part or a somatic part or a body cell. But then there are germ cells that are directly involved in reproduction, meaning they will form gametes. Clear? Now, let us come back to the gynoecium part. Now, remember, when the pistils, that unit of the gynoecium, when they are fused, like several pistils can be present and they'll be fused like this. Okay, they'll all be fused together. You can't separate one, two, three out. No, they're all fused. So when they are fused, we call it as syncarpus. Carpus refers to carpel. Syn means fusion. Okay, syn means fusion or fused. So syncarpus in this one, the first condition. Do you see how it is all connected at one area? It is like none of the free ends you can see. But when the pistils are free, meaning when they are not fused, we call it as apocarpus. Meaning, can you see here how these are all in the air?
Yeah.
How none of it is fused? So this is how a free, or you can say a free pistil or the apocarpus condition is that. So the first one, that is the syncarpus, this is found in papaver plant. And the second condition, that is apocarpus, it is found in Michelia plant. Okay, this picture is there in your NCERT book also. Now, talking about the pistil, just like how many parts did stamen have?
Stamen had two parts, two cells.
What were they?
The stamens are
Hmm. You are right. It has two parts.
the cells or the parts. It has
Parts. I'm asking parts. I'll draw it. Then can you say?
yes, pistil, filaments and anther.
Very good. Anther and filament are the parts of the stamen. Very good. Now, just like that, pistil also has three parts to it, right? There is stigma, style, and ovary. Stigma, style, and ovary, these are the parts of the pistil. Now, stigma, see here, this picture. See, this part that sort of looks like a base like this part is the stigma. The part that is long and continuing from the stigma is the style, and then there is the basal bulgy part which we call as the ovary. So stigma, style, and ovary are the three parts of the gyno or pistil. Now, stigma is the landing platform for pollen grains. So whenever the dehiscence of anther will take place, what is dehiscence of anther?
of anther.
Hmm?
Anther to release pollen grains.
Bursting. Very good. Yes. So once that will happen, So the pollen grains will be released. Now, if this is the stigma, so the pollen grain will come and land here. Okay. So that stigma is the landing platform for pollen grains. Then after that comes the style. Now, style is the elongated, means long, slender part. Slender means thin. It is the long and thin part which is present beneath the stigma, and it sort of connects the stigma to the ovary. And lastly, there is the ovary, which is the basal bulged part. Okay, it is basal means it is present at the base, and bulged means that it is swollen in nature. Okay. Now, inside the ovary, there will be present or the ovule. Okay, but before that, remember that ovarian cavity is present. Okay, and inside the ovarian cavity, placenta will be present, the placenta of the plant. And then the ovules, they arise from the placenta, meaning this placenta give rise to the ovules. So if we can say in a nutshell, inside the ovary, ovarian cavity is there. Inside the ovarian cavity, placenta is situated, and from the placenta, ovule arises. The other name for ovule can also be megasporangia. Just like how we had microsporangia, which was giving rise to microspores, same way, megasporangia, which will give rise to megaspore, which is the egg. What was the microspore?
It gives rise to four tetra.
Hmm. What is a microspore?
Spores are it is in pollen grains. And
Pollen grains, right? Microspores, right? They are only the structures that arise from microsporangia, isn't it? So the megasporangia, which is the ovule, it forms the megaspore, which is the egg. Clear? So remember these, the chronology, like how the basal bulgy part is the ovary. Present inside the ovary are ovarian cavity or locule. Locule means it's like rooms. Rooms are there inside it. Okay. And then inside the cavity, inside each of the cavity, there is placenta. And developing from the placenta will be megasporangia. If you say megasporangia or if you say ovule, it's the same thing. Okay. And then this megasporangia will give rise to the megaspore, that is the egg. Now, once the development of, you can say the reproduction of the flowering plant is completed, this ovule, you remember ovule, right? This ovule only will develop into the seed. So the fruits that we eat, they have a seed. For example, mango. Mango has that gutli. That is the ovule. Okay. And the fleshy part that we eat, that is the ovary. Okay. So when the like reproduction is completed, the ovary part, it starts getting to ripen up, and that ripens up and becomes the outer layer of the fruit. And then inside the ovary, we already know that ovule is present, and this ovule will develop into the seed of the fruit. Clear? So next time you eat fruit, you'll be thinking that, oh, I'm not eating fruit, I'm eating the plant's ovary. Isn't it? Just 1 second dear. Just 1 second. Yes. Okay. So any doubts up until now? Okay. Now you might see, if we talk about mango, how many ovules does the mango have? 1. How many ovules does a watermelon have? I can't hear you, dear.
Out of ovules.
Yeah, you can't count it, right? So
Yeah.
there is, like, you can say, a variation as far as the number of ovules inside the ovary goes. Okay. So there are many that have only 1 ovule in the ovary. That is in the case of wheat, paddy, mango, right? And then there are many ovules in the ovary, such as papaya. Papaya has so many seeds inside, watermelon, and even the orchids. Clear?
Yes.
Now talking about the megasporangia. Megasporangia, or you want to call it ovule, it's the same thing. Okay. Now the ovule is the structure which is attached to the placenta, By the means of a stalk. Obviously, there has to be something because of which the ovule would attach, isn't it? Now, like try to relate it with the human's placenta. The fetus in the human, they are connected to the placenta by which structure? Do you know? Even if you
Uh,
don't know the name, do you know the structure of it?
it's the umbilical cord.
Very good. Umbilical cord. So same way, here also, obviously it's plant, so it can't have umbilical cord. So it has a stalk, which is called as funicle. So the funicle is a structure that connects the ovule to the placenta. Now the region where the body of the ovule fuses with the funicle, we call it as the hilum. The body of the ovule where it is fusing with the funicle. And if we talk about hilum, so it is the junction between the ovule and the funicle. So this is the typical structure of the ovule. See, this is the funicle and this is how the ovule is attached to the placenta. Now, hilum is the junction between the ovule, this part, and the funicle. It's present right in the middle. Okay. Now, do you see these layers? One layer, two layer, right? Do you see these structures? So these are the integuments. Integuments are the protective layer. So there is the presence of an outer integument and then there is an inner integument. So two integuments are there that is responsible for protecting it. But the point where there is no integument, do you see how there is a point in the whole entire ovule which has no integument at all? So that point is called as the micropyle. Okay, it is sort of like a pore. Okay, so there is no integument present at one region which is called as the micropyle. Okay, this part. Now, micropyle is the small opening from where the pollen tube enters the ovule. Right now, just remember that it is the opening from where the pollen tube would enter. We'll see how that happens. Now, remember that whichever side micropyle is present, that side is considered as the top side. Even though it is pointed downwards, but this is the top hand side. And the basal part of the ovule is represented by the chalaza. Okay, chalaza is the structure which represents the basal part, meaning the base of the ovule, and the top of the ovule is micropyle. So opposite to the micropyle end is the chalazal end, and the chalazal end represents the basal part of the ovule, the base. Now inside the ovule, these structures, this one, there is a mass of nutritive tissue. It's a tissue which is very rich in nutrition so that it can develop, like it can nourish the developing ovule. Okay, so that is nucellus. Okay, it is a nutritive tissue. And the cells of the nucellus, they have abundant reserve food material, meaning they have so much of food storage that whenever the ovule or developing ovule would need it, it will provide it. Okay. Now, do you see inside this structure that has so many egg-like things? Where did the diagram go? Do you see this egg-like structure in which there are so many things? Right. This part is called as the embryo sac. Okay, this part is the embryo sac, and this is the part wherein the egg will be present. Imagine one egg is protected by so many layers, isn't it? That it does not get destroyed easily. So now coming on to the embryo sac. So inside the nucellus, embryo sac is present, which is the female gametophyte, because this embryo sac will contain the egg, that is megaspore. Now, an ovule generally has a single embryo sac formed from one megaspore. So inside every ovule, there will be one embryo sac. Is it clear?
Yes, ma'am.
All the parts of the megaspore is clear to you?
Yes, ma'am.
Okay, very good. Now you can note it down. Then we'll move forward. Also, dear, as far as the assignments and the test goes, what is the difficulty that you're facing? Are you like not able to submit it, or are you not able to access it at all?
whenever I open it, there is the topic given, the name of the thing we have learned is given, but there is nothing to, MCQs and all, nothing is there.
Okay. All
The
right. Oh,
I did in physics, there was, but after that, I have no questions there.
okay, so it's like complete, like the topic is there, the name
Yeah.
and everything is there, but
Yes.
it's blank inside.
Blank. Yeah. Whenever I try to submit it, it's nothing comes.
Okay, and have you ever tried to open it in Chrome, Google Chrome?
Google Chrome, I just click the link they have sent. They send this link, I click on it, but it opens in Safari for me, not in Chrome.
One thing, you actually copy it, like you press it and it copies,
Okay.
right? And
Okay.
then you open it in Google Chrome. Maybe it will work
Okay.
in that also. Okay,
Okay.
but
Okay. I'll try.
I'll let the team know about it so that you
Yeah.
won't face any problems regarding it.
Okay.
Okay,
Okay. Thank you.
yeah, you can switch off your video, write down something.
Done, ma'am. Done, ma'am. Done, ma'am.
Draw this diagram also, okay?
Okay, ma'am. Done, 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 ma'am.
All right. Any doubts up until now?
No, ma'am.
Okay. Now moving forward, let us talk about megasporogenesis. Just like how we had microsporogenesis, same way we have megasporogenesis with a little bit of differences according to the division. Now we know, just like how microsporogenesis was the formation of microspores, same way megasporogenesis is the formation of megaspore. Now, the process of formation of megaspore from the megaspore mother cell is called as megasporogenesis. Now, the megaspore mother cell is differentiated by the ovule in the micropylar region, meaning this cell structure is present in the micropylar area. Now, the megaspore mother cell, it is a large cell which contains a dense cytoplasm and a prominent nucleus. Prominent means a very easily visible nucleus. Okay. Now, what happens is that the megaspore mother cell, it starts to undergo the process of meiosis to form haploid megaspore. 4 haploid megaspores are formed. Now, the thing is that out of the 4, 3 of them are useless. They degenerate and only one is functional. Okay. And the functional megaspore develops into the female gametophyte, that is the embryo sac. Okay. And this method of embryo sac formation, because only one one megaspore is performing in it, so we call it as monosporic development. Mono means one. Sporic refers to the one megaspore that is performing. So we call it as monosporic development. Because out of the 4 haploid megaspores, 3 of them degenerate. Only one is functional, and that is why. Clear? Okay, now note this down and we'll keep it till here only.
Done, ma'am.